Hey guys, today I will be explaining to you the function of the FGFR2 protein, but focusing more specifically on the dysfunction and why the change in this function causes uh, Cruzon syndrome. Okay, as we know that there are about 50 known mutations of FGFR2, and these mutations are what causes Cruzon syndrome to occur. 95% of these mutations are because of a missense mutation, meaning that the amino acid sequence that we were looking at in previous pages, in, for example, protein structure, um, changes one of those amino acids to another one. For example, a 340 cis 342 arg is the name of one mutation which changes the cysteine in the 342 position to arginine, and that's this fellow right here. Okay, we know from before that this amino acid sequence is vital to how the how the protein is going to fold and how it'll therefore function. Because so if we take this mutation into consideration, okay, this change occurs in the Ig3 protein domain. Okay, what is a protein domain? A protein domain is basically any structure of the protein that can function on its own or is vital for the function of the whole protein. So when we do that change, this amino acid change from cysteine to arginine, this causes that conformational change to occur in that protein domain, right? The protein domain Ig3 goes from amino acid positions 256 to 348, and because it occurs in that protein domain, it will affect the functionality of the Ig3 domain. Now, having a look at figure 6, we have our normal FGFR2 protein functioning over here on the left and our mutated one on the right. So here on the top we have our signals, or it is our fibroblast growth factor that acts as our signals, or a ligand as we can call it, that approaches the receptor and is then activated, which then causes signal intracellular pathways to occur, and then goes on to proliferate, cell death, and for a number of reasons that we had stated before in our previous pages. But because of this change here, what happens is it causes a conformational change in this Ig3 domain, which means that this ligand consistently activates this tyrosine kinase domain, which causes the intracellular pathways to be constantly activated, which leads to extended proliferation or extended cell death for an exceeded amount of time that it is not required.